Hi, this is the second of 3 lectures to make up for our snow day. And this one is on recombinant DNA and biotechnology. So this is a general outline that will cover both of the 2 remaining lectures. And in this one, I'm going to be talking about the discovery of restriction endonucleases and what they're capable of doing. And then we're going to talk about how we can use restriction endonucleases as a mechanism to analyze genetic structure. And we're going to talk about several of the different mechanisms that geneticists use to understand and describe the genetic structure of organisms. The second part of this will be on actual genetic engineering techniques to try to use restriction endonuclease technology to actually manipulate the genetic structure of organisms and introduce genes from one organism into another. So let's start by talking about restriction endonucleases. So these 3 individuals, Werner Arber, Daniel Nathans, and Hamilton Smith, all won the Nobel Prize in 1978. And they won that prize for discovering restriction endonucleases. And this was based on research they did in the 50s and the 60s. And they weren't out trying to create an industry. They weren't out trying to develop a whole field of biotechnology. They were simply trying to better understand how phages interact with bacteria cells. Now, we've talked about the biology of phages in the past. A phage is a virus that's specialized in attacking bacterial cells. So it attaches to the cell, and then its DNA will enter that cell, and that triggers the degradation of the host cell's DNA, the bacterial DNA. And the viral DNA, the phage DNA, then goes through replication and transcription to produce the viral proteins. And so the viral DNA then is assembled in those viral proteins to make new phages. The cell bursts or lyses, and that releases the new viruses, the new phages that can now attack other cells. So this is the typical life cycle of a phage. And phage and bacteria have been co-evolving for forever, for millions and millions of years. And this looks a little one-sided. It looks like all bacteria are going to suffer if they're exposed to phage. But it turns out bacteria have evolved mechanisms to try to resist phage attack, because any bacterial cell that can do that is clearly going to be favored by natural selection. When biologists study phages, you can see the effect here. And so this is part of a petri dish. And what researchers have done is put bacteria on this plate so that it will grow into a thick lawn of bacteria. So all the lighter colors represent regions on this petri dish where bacteria are growing. And then they introduce the virus. And the virus goes through its life cycle, and it causes eventually the cells to burst. And it produces these clear dark areas that are called plaques. So a plaque is simply what's left behind when the phage has successfully gone through its life cycle and destroyed the cell and reproduced as it did that. And these plaques then increase in size as the phages move out into the existing colonies of bacteria. And eventually, these plaques can increase in size until all of the bacteria on that petri dish have been destroyed. But again, well, when we study these, what researchers will do again is create a colony of bacteria on a plate. So in the upper part of this picture, it's a diagram of a petri dish that has lots of E. coli cells. All little dots represent E. coli cells. And these are all described as cells that are T1 sensitive. T1 is a particular kind of phage, and these are cells that are susceptible to attack by T1 phage. And so you expose them, then you transfer them to other plates and expose them to the phage, and what happens is that the phage successfully attacks the cells lyse, and they all die. But there are a few cells that may have had mutations that make them T1 resistant. So they can survive the attack, and these are shown on the 3 plates in the bottom of the slide in red. So those are all colonies of cells that are derived from a single cell that had a mutation that made it T1 resistant. And they've survived and produce offspring and are not affected by the presence of the phage. So what these 3 researchers are doing is try to figure out how these cells do this. What kind of mutations did they undergo to be able to change from being T1 sensitive to T1 resistant? And what they discovered was these cells all produced a group of enzymes, proteins, that are referred to as restriction endonucleases. And that term comes from the fact that any bacterial cell that can produce these enzymes can restrict the life cycle of the phage, essentially prevent the phage from successfully destroying the host bacterial cell. And they're also called endonucleases because what they do is that they bind to the DNA of the phage somewhere in the middle of the molecule, and they snip that DNA. They essentially cut it up into little pieces, rendering it non-functional. And then the bacterial DNA, the host DNA in the bacterial cell will continue to do what it's supposed to do. The viral DNA eventually is degraded. So in this diagram, the little green dots represent the restriction endonucleases that are being produced by the bacterial cell that attach to the viral DNA as soon as it's entered the cell, cut it up into little bits and destroy it, and the host cell survives. So if a cell can produce those restriction endonucleases, it then becomes resistant to attack by phages. And that's what happened to those resistant cells on the previous slide. So that's all really good and interesting basic research. I mean, they went into this just trying to better understand what happens when cells evolve resistance to phages. What goes on, what's involved in the mutation, what are they producing, and how do they affect this kind of resistance? And so it was all just basic research, trying to understand a little bit more about the relationship between phages and bacteria. So they didn't do this work, again, with any intention of creating a whole biotech industry. But that's what happened. And the reason that happened is because of the specific ways in which restriction endonucleases go about their business. So what a restriction endonuclease will do is bind to DNA, and it binds at a particular sequence of bases that's called a recognition site. In this case, we're dealing with restriction endonuclease that's called EcoR1. Eco stands for E. coli. That's the cell that produces it. And R1 just means it was the first restriction endonuclease that was identified from E. coli. And what this particular enzyme will do is seek out a particular base sequence. In this case, the sequence is GAATTC. And this sequence has an interesting quality, an interesting characteristic in that it's a palindrome. A palindrome is any sequence that can be read in the same way backwards and forwards. So if you look on the upper strand of DNA at the recognition site, you see the sequence GAATTC, and in the lower strand, you see the same sequence, GAATTC, in the 5, both of those being read in the 5' to 3' direction. So that sequence of 6 bases is a palindrome, and that's the sequence that EcoR1 binds to. And then it cuts the DNA between the G and the A bases on each strand. And when it does that, it cuts the DNA unevenly. So there are these little tails of single-stranded DNA that are hanging out. In this diagram, they're called complementary tails. They're also referred to as sticky ends. Because those bases are single-stranded, they want to bind with another DNA. They want to form hydrogen bonds, and they want to become double-stranded. And since there is a sequence of single-stranded DNA on the other piece that's been cut, there's a tendency for these to want to join back together. And that's why they're called sticky ends. They tend to stick together with other pieces of DNA that were also cut by the same restriction enzyme. So that's an interesting discovery of how these restriction endonucleases work. And what that means, well, lots of other restriction endonucleases were discovered, hundreds, maybe even thousands. So here are 4, and they're named based on the species of bacteria that produces them. So the first one is called Hindi, and it's produced by the bacteria Haemophilus influenza. And the second one is BAM, H1, that's produced by Bacillus amyloquafaciens, and so on. And hundreds of these have been discovered, perhaps even thousands, all produced by different bacteria, all for the same purpose of binding to phage DNA when a cell is infected by that phage DNA, and then cutting it. And when they studied these different restriction endonucleases, they discovered that they all cut at different sequences. So Hindi cuts at AAGCTT, and BAM-H1 cuts at GGATCC, and so on. Some of these produce sticky ends, like those first 2, and others are called blunt end cutters or blunt cutters that don't produce sticky ends, but cut the DNA, again, at a particular site. And so by understanding how these restriction endonucleases function, researchers then realized that they could take advantage of the characteristics of these enzymes and use them to manipulate, to cut DNA in particular places for particular purposes. So basically, if we want to use this for biotechnology, to create recombinant DNA, we simply cut 2 pieces of DNA with a restriction enzyme, in this case, EcoR1. It's going to produce sticky ends. And then those sticky ends of DNA will join together. We have to add a little ligase to make that happen. But it doesn't matter where the DNA comes from. If you cut DNA with the same restriction enzyme, then some of the DNA molecules from one organism could combine with some of the DNA molecules from another organism to produce a molecule of DNA that is now recombined, that is actually consists of DNA that came from 2 different organisms. And that's the basic way in which recombinant DNA and biotechnology can work. But before we get into how we can use this to create new kinds of organisms and move genes from one organism to another, I want to talk a little bit about how we can use these restriction enzymes to study the genetic structure of the genomes of organisms. And one way we can do this is by doing a procedure that's called a restriction fragment length polymorphism. It's called a RFLP. And what a RFLP is a demonstration that in different organisms, there can be a difference in the restriction sites that are present. And therefore, there are differences in the way in which DNA can be cut. So imagine we're looking at 2 different strains of flu, and we've got strain A and strain B in this diagram. And those viruses all have a DNA of 1,000 base pairs. Most viruses are bigger than that, but let's just assume that the DNA for that flu virus has 1,000 base pairs. And we want to know if they're the same strain. So what we can do is expose each of those strains to a particular restriction enzyme. In this case, it's, again, EcoR1, which is going to seek out the sequence GAATTC. And what happens in strain A is that there are 2 of those restriction sites present in that DNA. So it cuts it twice and to produce 3 pieces of DNA. But the bottom strain has a mutation in one of those restriction sites. So you look at the restriction site on the right in DNA molecule B, you see that the sequence is GAATCC instead of GAATTC. So EcoR1 will not recognize that 6-base sequence, and so no cut occurs there. But the GAATTC sequence in strain B is still in the same location as it is in A on the left. So that particular piece of DNA is cut only once into 2 pieces of DNA. So you expose your sample of DNA to the restriction enzyme, it cuts the DNA, and then you take that DNA and you put it into an electrophoresis gel. You run that gel out and it separates the pieces of DNA by size. And we can see the 2 lanes for samples A and B in the diagram on the right. The third lane is for a series of size markers that we can use to estimate the size of the pieces of DNA. So we're just putting in pieces of DNA of known size and spreading them out. So we can see that sample A results in the production of 3 pieces of DNA that are 500, 300, and 200 base pairs long. And DNA from strain B produces only 2 pieces of DNA, one that's 700 and one that's 300 base pairs long. So the difference between these 2 strains of flu virus is an example of a polymorphism. A polymorphism is a term we use to describe any traits that differ between organisms. And in this case, these 2 strains of the flu virus differ based on the sequence of bases in that second restriction site. So that EcoR1 cuts in strain A, but it doesn't cut in strain B. So we can see that strain B is genetically different from strain A. And these kinds of analyses can be used to identify particular strains of a flu virus. And that's one of the techniques that are used to identify the particular flu viruses that are coming around and causing disease. So RFLP is one of the first techniques that can be used to analyze the genetic structure of an organism. Essentially what you use it for is to create what's called a restriction map. And a restriction map is simply a description of where the restriction sites are on a particular piece of DNA. And one technique to do that is to extract the DNA. And so we've got here a 7,000 base pair piece of DNA, 7.0 kb means 7,000 base pairs. And we're going to cut it with restriction enzymes. And we've got two different enzymes that we're going to use. We're going to use Hindi and we're going to use SAL. So first we're going to run some DNA that's uncut, just kind of as a control. And then we've got a lane where the DNA is cut with Hindi. And then we got a third lane where it's cut with SAL. And then we've got a fourth lane where it's cut with both. And by looking at the fragments of DNA that are produced in our electrophoresis gel as a result of the action of those restriction endonucleases, we can put together a map of where the restriction sites for these two restriction enzymes are located. Now, just based on the raw data that we see in the electrophoresis gel, there actually are two possible genetic maps or restriction maps that can be drawn. Each of these enzymes is cutting the DNA in one location and producing two pieces of DNA. The fact that they're of different size indicates that it's cutting the DNA at a different location. And model 1 on the left shows the cutting sites being both close to the left end of our piece of DNA. And model 2 on the right shows the Hindi restriction site to the left of our DNA molecule and the SAL site to the right of our DNA molecule. Each of those models predict the sizes of the pieces of DNA that you would get. So the model 1 predicts that we would get a piece of DNA that's 4 kb, a piece that's 8 kb, and a piece that's 5.8 kb. The model on the right, model 2, predicts that we should get a piece of DNA that's 1.2 kb, a second piece that's 5.0, and a third piece that's 0.8. Now, when you look at the combined cell, the lane on the right, where they've been cut with both enzymes, you see we have 3 pieces of DNA that match up really well with model 1, the model on the left. So that tells us where these particular restriction sites are located. And that's a restriction map. And a restriction map is a useful tool to describe the structure of a particular piece of DNA. And again, we could imagine that this is a piece of DNA that's coming out of a virus. So now we have a tool that we can use to analyze the structure of that DNA molecule by creating a restriction map. Different strains of a virus might produce a different restriction map because there have been mutations in its DNA that either add or take away restriction sites. So producing a restriction map is a very useful tool in understanding the structure, the genetic structure of an organism. So this slide is another example of how we can, there we go. It's another example of how we can create a restriction map. Now we're doing essentially the same thing. We're using 2 different restriction enzymes. We've got a piece of DNA that is 750 base pairs long. So we cut it with enzyme A on the left, and we get those 5 pieces. We cut it with, so there are, 4 pieces of DNA with 3 restriction sites. We cut it with enzyme B in the middle, and we get 3 pieces of DNA indicating 2 restriction sites. And then if we cut it with both, do the double digest, we get those 4 pieces of DNA. Now, what we've added into this diagram that wasn't in the previous slide is those particular pieces of DNA indicated in red with the red asterisk have been, the original piece of DNA was marked, a marker was put on the end of the piece of DNA. Usually as some sort of radioactive marker, perhaps. So we know that the piece of DNA that's 200 base pairs long and the piece of DNA that's 100 base pairs long had to be at the end of our original 750 base pair piece of DNA, and the 40 and 450 base pair pieces of DNA are in the middle. And then when we digest with enzyme B, we see that there are 3 pieces of DNA, and the 250 base pair piece of DNA is in the middle. And then we get the double digest that's on the right. Again, there are 2 possible restriction maps that can be drawn based on this data. And the actual one is up on top where we get, it indicates where the restriction sites are for enzyme A and the restriction sites are for enzyme B. And that gives us the pieces of DNA in the double digest that we would predict based on that arrangement. If you look in the hypothetical alternative, you can see that would end up producing different sets of pieces of DNA of different sizes. So it's not consistent with our double digest, and the upper diagram is. So again, we've created another restriction map showing the restriction sites for these two enzymes. And again, on this slide, you can see the pieces of DNA that are indicated in red, the 200 and 100 base pair pieces of DNA are the ones that have had a radioactive marker added to the end of the original DNA molecule to show that those are pieces of DNA that are on the ends of our original piece. Now, this technique of doing a RIFLIP analysis works pretty well when we're dealing with the small genome. Anything under 10,000 base pairs or so, 10 kb, works pretty well. But eukaryotic organisms have billions of base pairs. The human genome has 3 billion base pairs. So if we simply cut it with a restriction enzyme, which is what they're doing in the upper left of this diagram, we've got 3 different samples of DNA. They're cutting it with a restriction enzyme and then running it on an electrophoresis gel. So they're using 3 different kinds of enzymes. You get really what amounts to so many bands that really what it looks like is a smear of DNA rather than distinct bands. So it often is not very useful as creating a process of genetic markers. So the technique that's illustrated in this slide is something that's called a Southern blot, because it's called a Southern blot because it was developed by a man named Southern. And then the first step is to transfer the DNA from that electrophoresis gel onto a piece of filter paper. And you do this by putting it on that apparatus in the picture. So you've got a tray filled with a buffer, and a sponge is sitting in that tray in the buffer solution, and it's going to draw up the buffer, and it's going to travel up through the gel. And you've also placed a piece of filter paper on top of the gel, and then a bunch of paper towels on top of that to absorb the buffer as it moves up, and then a brick just to hold it all in place. And what happens is that the buffer will absorb the DNA from the gel, transfer it to the piece of filter paper, and the DNA stays behind on the filter paper as the buffer travels up into the paper towel. So this is a process simply to transfer it to filter paper so it can be more easily manipulated. So now what you have is a piece of filter paper with DNA spread out in each of those lanes based on the size of the DNA. But again, there's so many pieces of DNA that it doesn't provide any useful information. So what we're going to do is create what's called a radioactive probe. And this is a probe that consists of a small piece of DNA with maybe 20 to 30 base pairs that has a radioactive marker attached to it. And we're going to wash it over the piece of filter paper that has the DNA. And any place where that DNA in the radioactive probe can find a complementary sequence, it will bind to the DNA in the filter paper and be attached to it. So we simply let the radioactive probe, or expose the filter paper to the radioactive probe, let it sit there for a while, then wash all the radioactivity off. And the only radioactivity is left behind is radioactivity that's attached to the probe that has bound to a particular band of DNA, because the sequence of DNA in the probe is complementary to the DNA in the band. And so that gives you a much smaller number of bands. And again, you can look at those bands and measure the sizes of the pieces of the DNA and use that as a study of the structure of the DNA for that particular source organism. This is something that's also called DNA fingerprinting, because when you do it, you can compare pattern of bands that are produced. And this is something that's often done in a crime investigation. So you've often seen on shows like CSI where they talk about the DNA evidence and they have a DNA match. So on the right is kind of an example of a Southern blot, a DNA fingerprint, in which a sample of tissue or blood was taken from the victim. And also from a specimen. Let's say this victim was raped and they managed to collect a specimen of semen that was deposited by the rapist. They can do this RIFLIP or this Southern blot analysis and create a particular banding pattern. And then if they have a number of suspects, they can collect a sample of tissue or blood from those suspects, do the same procedure, and then see if the bands match. And I think you can easily see that among the 3 suspects, suspect number 1 has a banding pattern that is identical to the banding pattern from the semen specimen that was taken from the victim, the rape victim of this crime. And so that's pretty strong evidence that suspect one might be the rapist. It doesn't prove that he raped the victim, but it does prove that his sperm was deposited in the rape victim's body. So that's pretty strong evidence that he perhaps is guilty of rape. And this is the kind of evidence that is used to either convict or, in many cases, acquit individuals of a charge. There are many individuals that have been serving long prison terms because they were convicted of rape, and then the DNA evidence that was analyzed later showed that there was no match between the specimen of semen collected from the victim and that particular convicted individual. And so many men have been acquitted and released from prison as a result of this kind of evidence. Okay, a second technique that we can use, or another technique that we can use to describe the structure of the genome of an organism, is to sequence the DNA, to actually determine the sequence of all the bases in a sample of DNA. And this is done by using a modification of a nucleotide. The nucleotide that's in the top part of this picture is a deoxynucleotide. It's a typical DNA nucleotide. It consists of a base, the deoxyribose sugar, and 3-phosphate functional groups. The one below is called a dideoxynucleotide, and that's because it's missing a second oxygen. Deoxynucleotide is missing one oxygen, the one that would be attached to the hydrogen at the bottom of that sugar molecule. The dideoxynucleotide is missing both of the oxygens. So dideoxy means two. It's lacking two oxygens. And that dideoxynucleotide will enter into the DNA replication process, but because it's lacking an OH functional group on the number 3 carbon, new nucleotides can't be attached to it. So the DNA replication process essentially stops whenever a dideoxynucleotide is inserted as a complementary base pair when DNA is being replicated. So how do you use this to sequence DNA? Well, you start out with a template strand of DNA and provide all the nucleotides that they need in order to go through the process of DNA replication. And that happens by adding the nucleotide to the 3' end of the growing strand of DNA. So that's the typical process of DNA replication. But if a dideoxynucleotide is included in that mix, then occasionally a dideoxynucleotide is going to be attached instead of the normal deoxynucleotide. So in the example that's shown here, what we have is a piece of DNA that's being extended in the 5' to 3' direction. And you see in the first example, the bases C, T, and A were added. And that third nucleotide that was added, the A, is a dideoxynucleotide that's indicated by the little asterisk. And so when that happens, then no more extension of that single-stranded piece of DNA can occur. So that ends the process of DNA replication for that particular piece of DNA. In other pieces of DNA in this sample, a normal A may be attached in the third is the third nucleotide, and then it goes on to the 5th where another A is supposed to be attached, and that one is a dideoxynucleotide, which ends replication. And so what you get when you do this are pieces of DNA of different sizes, and based on the size of the piece of DNA that's produced, then in each of these cases where DNA replication was truncated, was stopped, that tells us the position of that particular nucleotide. And we can do this for a dideoxynucleotide T and a dideoxynucleotide C and a dideoxynucleotide G, and that tells us the size of that piece of DNA. So here's a typical kind of gel that we might get, and you just read up from the bottom and you can see that the first nucleotide is a C and the next one is a G, and then a C, and then 3 T's, and then another C, and so on all the way to the top. So this technique determines the sequence of the bases of DNA in that particular sample of DNA. So here's just another slide that kind of shows how you go through that process. And to make this work easier, you simply attach a colored marker to each of the different dideoxynucleotides. So you've got red for A and blue for C and green for G and yellow for T, and put them all in the mix. And then you look at the different size pieces of DNA, and each one is going to end with a particular color. And so what color you see will determine which base is in that particular position. So this is a process that's become automated, and you get a printout like this, and all of these are 4 different colors corresponding to the 4 different kinds of dideoxynucleotides with a color marker attached to it. And so you simply look at the order of the colors in each of these. These can actually be read by a scanner, so it's all automated. It can be done fairly quickly, and we're now capable of sequencing large quantities of DNA. When this process started 20 or 30 years ago, they could do 100 nucleotides a day. It was a pretty slow process. Now you can do thousands, tens of thousands, maybe even millions in a day. And so the rate at which DNA can be sequenced has gone up dramatically. So the black line in this diagram represents the number of human genomes that have been sequenced since this first started around the year 2000. And that's going up exponentially. And the reason it's going up is because the speed of this process is going way up. And because the speed is going up, the cost is going way down. So used to cost, the first DNA, human DNA that was sequenced cost something on the order of $3 billion. Now you can sequence a human genome and the cost is less than 1,000, and it's going down. And pretty much it's going to be so easy and so cheap to sequence DNA that it's going to be part of a routine medical exam. When you first go in to see a new doctor, to take a sample of tissue or blood and sequence your genes because it's so cheap and so easy to do, and there's a lot of information. So this is kind of a sequencing revolution that's occurring, and that's why there's so many companies out there now like 23andMe that for less than $100 you can get a lot of genome sequence information. It's not necessarily a complete sequence, but at least enough to know things like your genetic heritage, whether your ancestors came from Europe or Africa or Asia or wherever. All that information can be gleaned out of the sequence data that they get. So sequencing has become routine. Another mechanism for analyzing the structure of DNA is something that's called a polymerase chain reaction. Again, this is taking advantage of and utilizing the DNA replication process, but in a slightly different way. So what you do is take a sample of DNA, and then you want to denature the DNA. That means you want to separate the 2 strands. So in this particular diagram, starting on the left, we've got a double-stranded piece of DNA, and we're going to denature it, separate it, so that we have 2 single strands. And then that little piece of green that's attached to each one of those is called a primer. And a primer is a small piece of DNA that will attach base on base complementarity. And then we expose that to DNA polymerase, and the DNA will replicate. And it essentially replicates the DNA between the 2 points of attachment of the 2 primers on each of the pieces of DNA. So we now have a new double-stranded piece of DNA. We've doubled the amount of DNA that's present. And then we go through the same process. We separate the DNA, denature the DNA, separate each DNA molecule into a single-stranded molecule, attach the primer, and then replicate the DNA between the primer. So each cycle replicates the DNA molecule. So we're getting a doubling of the number of DNA molecules with every cycle. So very quickly we can produce 1 million or 1 billion. If we do it for 20 cycles, that's going to give us 10 to the 9th copies of DNA. So we can get an awful lot of DNA, and DNA that's readily visible on an electrophoresis gel. And this is something then that can be used to analyze the structure of the DNA from where that sample came from. If you look at the link on this cell, on this slide rather, there is an animation that you can go to see an animation of this process. It might make it a little easier to understand. So polymerase chain reaction now has also become automated, and we use equipment called thermal cyclers. And a thermal cycler simply takes a sample that you prepared and then changes its temperature. So the first thing you have to do is raise its temperature to about 95 degrees in order to denature the DNA, separate the 2 strands of DNA. Then you drop the temperature to about 55 degrees, and that's a temperature in which the primer can anneal to the DNA. And then you raise the temperature up to about 72 degrees Celsius, and that's the best temperature in which DNA polymerase can work to replicate the DNA. Then you just repeat that cycle over and over and over again. And you can program the cycle into the thermal cycler machine. You let it go for a few hours, the DNA is replicated, and then you can spread it out in electrophoresis gel like this, and that gives you some important information about that DNA. This particular slide is about an analysis that's called microsatellite analysis. And microsatellites are little pieces of DNA in which 2 to 4 base pair sequences are repeated over and over again. And because you've got these repeated sequences, you often get errors in DNA replication. So these are sequences that tend to be polymorphic. They tend to differ a lot among organisms. So in this particular slide, we're showing an electrophoresis gel with about 15 different individuals, and they differ in the size of a particular piece of DNA. And we identified that piece of DNA by designing a primer that would attach to the DNA on either side of these repeated sequences. And it turns out that some of the individuals have 198 base pairs in this particular piece of DNA, and some have 176. And some individuals have both. So think of this as a gene, as a marker for a gene. The first lane has an individual that's homozygous for the 198 base pair piece. The second lane is an individual that's homozygous for the 176 base pair piece. And the third individual shows an individual that's heterozygous, that has one of each of those on its 2 chromosomes. And so this is a way in which you can fairly quickly do a genetic analysis, and this kind of information can be used to create individual identities or look for paternity or all sorts of interesting things about all sorts of different organisms. And it's based on polymerase chain reaction. Okay, so I'm going to end this particular YouTube video now because we've talked about how we can use restriction endonucleases and biotechnology to analyze the structure of DNA. And the next set of slides, we're going to study how we can use restriction endonucleases to actually manipulate the genetic makeup of organisms.